Welcome. I'm Andrea Manole. I'm a senior scientist at iPSCs Biotechnologies. And today I'm going to talk to you about our work on developing a co-culture system between dopaminergic neurons and astrocytes from human iPSCs. So the dopaminergic neurons, you can find these images on our website. They are fully differentiated. They are very compatible with a wide range of cell-based assays, and they can be cultured very long-term. The same idea is with the astrocytes. They're very highly pure, and they're compatible with a lot of assays, which I'll talk about later on. So the schematic, the way we perform our experiments is we defrost the dopaminergic neurons, we culture them. In the first 7 days, we observe the presence of midbrain markers, and we're able to, in the first 7 days, co-culture them with glia. 2 weeks in, we're able to observe calcium activity, and later on, more mature markers and also MEA activity. The astrocytes can also be defrosted similarly, and in the first 7 days, we observe these astrocyte markers. We're able to co-culture them, and as they become mature, they also release cytokines. So on our website, you're going to be able to find images like these. When we defrost the dopaminergic neurons, we are able to see that they are very, very highly pure, and also there's a high viability. They also express TH and FOXA2 for dopaminergic markers and MAP2 for neural markers. And that's great. Of course, whether they are functionally able to release dopamine, that's something we have to test. That's very important to reflect more in vivo settings. So together with a collaborator that developed a cool technique that involves afterburners, modified nanopipettes. What she was able to measure in our media was dopamine in the picomolar range, and she has a lot of controls to show that the dopamine sensor was specific to dopamine. Then I'm going to play 2 movies. This is calcium activity of our dopaminergic neurons, and this one is for our glutamatergic neurons. And then here we extracted several measurements, including event frequency and maximum peak amplitude, and compared them with the competitor cells, and our cells outperform the competitor cells in essentially all measurements. The astrocytes also have high viability upon thawing, and they express the maturation markers, GFAP, 100 beta, and other markers. And functionally speaking, upon stimulation with IL-1 beta, they release cytokines, including IL-6 and IL-1 beta, more or less to the same level as the primary astrocytes. And this is a more complicated experiment where we defrosted astrocytes and we cultured them, then we stimulate with IL-1 beta. We wash out the cytokines after 5 hours, add back in neuronal media. In 24 hours, we collect that media and grow our dopaminergic neurons. And what we're able to see over 7 days is that the neural length of the dopaminergic neurons that was grown in the IL-1 beta previously stimulated cells was reduced. And here on the top, I'm just showing that there's expression of IL-6 and IL-8 cytokines in the media. The astrocytes can also be cultured with neurons, like I said before, and on an MEA plate, we can start to see if there's any difference in measurements if we culture the neurons with iPSC-derived astrocytes. And indeed, as you can see, the mean firing rate within 5 days was significantly increased in the neurons with iPSC-derived astrocytes, as well as with the primary astrocytes, and it was to the same, more or less the same level. And also the synchronous activity was significantly higher. The presentation will be available to download soon. Thank you, and I hope to see you in person at the poster session.